My name is Thierry Zoré. I did my PhD in France in the Institute for Structural Biology in Grenoble, where I learned crystallography, X-ray crystallography, on valence factors. Then I moved to Cambridge in England, where I got to learn cryo-electron microscopy. And with this knowledge in hand, I joined the group of Max Kreil at Monash University in 2016 to work on non-ribosomal peptide synthetase. So this non-ribosomal peptide synthetase, also called NRPS, are big machineries. They work as assembly lines, and they are responsible for the production of some of our last resort antibiotic, like teicoplanin and vancomycin. And it's very important to study them because we believe that if we understand how these machineries work, we can then modify them and produce new compounds that will help us to fight bacterial resistance in the future. So my project in this lab is to understand how these machineries are assembled together, and they're actually made of several modules, and at the moment it is not known how modules interact with each other. So being a structural biologist, I try to understand what makes this machinery work and how we can modify them or change these little modules to force them to create new compounds that could be used in the future for antibody therapy. And one of my projects was to work on very rare eukaryotic NRPS, because most of the NRPS are expressed by fungi and bacteria, and they are very rare in eukaryotes. However, there are some instances where eukaryotes use these NRPS machineries. And if the final goal is to modify these machineries, every new type of domains we can use to modify these machineries is important. And the fruit fly expresses one very interesting domain that is included in this NRPS. It's very fast and it's very active. However, until now, no one knew what was this domain made of. So what I did, I used X-ray crystallography to solve the structure of this domain. And this is very important because, as I said, when we know what makes these machineries, what these machineries are made of, then we can intervene domains and create new machineries that will help us develop new drugs. So specifically, I express proteins, purify proteins, and try to force them in making crystals. So a crystal of protein is an assembly of the protein. They pack each other in a lattice, and that's a crystal. Then we can use this crystal in a synchrotron, and with that we will have diffraction data, and this diffraction data can be processed into what was the structure of the protein forming the crystal. So the final goal of my research is to understand how these big machineries are assembled. So each module needs to interact with a different module. And at the moment, if we try to modify these assembly lines, then the new machinery mostly fail. It doesn't work. And the reason is that we don't know how different modules in the machinery needs to crosstalk. So another of my project is to basically understand how different modules can connect with each other, and then what is important to keep between different modules so that the machinery is functional. And at the moment, this is not known. One of the reasons is because most of these NRPS machineries are produced by bacteria and fungi that grow at relatively low temperature. So their proteins, or the machinery, are actually very flexible and very easily denaturated by heat. So when we try to work on them, the machinery is not in a good shape, and we cannot study it by crystallography or electron microscopy. So I recently secured a small grant from the CAS Foundation which stands for Contributing to the Australian Scholarship and Science. They helped me kickstart a new project where I'm actually going to work on a thermophilic organism that produce a machinery that is very stable at very high temperature. So with this, we hope that we will finally gain the missing knowledge to understand how a machinery is built. And with this knowledge, we will be able to modify the machineries that produce the last resort antibiotics that we need so much. So this is really important, and we can't thank enough the CAS Foundation for helping us with this project.